Can people hear me? Excellent. Thank you for the back of the room. Dramatic lighting, too. I love that. Thank you so much. It's really an honor to be able to come and be with you today and to talk with you all. I was just saying earlier that one of my favorite parts of this gig as being a professor is being able to go out and meet scientists of all levels and to talk about their science, get to know them all, and then also to talk a little bit about the work that my folks do in my lab. I said that they have all the fun now. I get to go and travel and talk about all the work that they do. Before I get into that, I just wanted to tell you just a little bit about the ASM Distinguished Lecturer Program. And this is going to be interesting because I don't know exactly where to look. There's so many screens here. This is a beautiful campus and a beautiful reading space. So kudos to you all for that. So a little bit about the ASM Distinguished Lecture Program. So it's a program that's been going on for over 60 years now. And it's a program that allows lecturers like myself to come in and talk to you all and share our work. And it's sponsored in part by ASM and the Waxman Foundation for Microbiology. And just briefly, a lot of this was already mentioned before. There's so many opportunities and there's so much about ASM that's wonderful, right? It's our professional society. And it allows me to come to talk with you, right, and network, right, with peers and share all the exciting microbiology, to understand that microbiology is all interconnected, right? There's a lot of themes that transcend your one model organism, and that's really cool to discover. There's a lot of career development opportunities, right? And opportunities for travel awards, right? For funding to come to these meetings, to learn about the science, to network, right? And also to build community, right? Community is so important, right? If we can't all get along and we don't know each other and work together, then what's the point, right? So it allows us to get together. It allows us to become ambassadors for science, teach some people that are not well informed about science what should be known, and help shape policy as well. And this can be done on all different levels, whether it's your local branch or even at the national and global levels too. And so the last thing is, if folks at ASM headquarters always want me to tell you a little bit about why I'm an ASM member. And so I've been an ASM member since I was a graduate student at the University of Wisconsin. And I remember my PhD advisor coming in and he's like, man, you've got to be part of our society, right? There's so many great benefits there. And so I became a member in 2003, and I've never looked back since then. So it's been over 20 years now, right? My first publication was in the Journal of Bacteriology, and to this day, we still publish in Journal of Bacteriology, right? And it's important that you contribute to your society journals, right? And why, right? People are, why do we do that? Why not go to the other journals? And the reason why is so I teach undergraduate biochemistry, and I have some business students, and they've been teaching me a little bit about return on investment. And it turns out that there's definitely a big bang for your buck, right? Your membership dues and publication charges come back to you, right? In the programming, like the ASM Distinguished Lecture Program, like those training grants, like the Peggy Cotter Award that we heard about, right? So there's all these great reasons why to be a member and to be an active participant, right? And for me, ASM has brought me up as a scientist, right? I've attended and done workshops on communicating science at ASM headquarters. It's one of the benefits of being in Washington, D.C. And the other is that I was a participant in a program that's had many names over the years, the ASM Summer Institute, which then became known as the Cadner Institute, in memory of Bob Cadner at the University of Virginia, who was an active mentor and participant in that. I learned how to give a good talk, and I learned how to write, proposals, right? And these are all things that, again, ASM helped me to do. So please, if you're not a member already, please consider being a member, both at the branch level and at the national level. Okay, I want to make sure I stay on time. And I wanted to tell you a little bit about some of the work that we've been doing to understand how Staph aureus decides to turn on the production of toxins and other virulence factors. Okay, and so again, this is a story that is it's a group effort by folks in my lab, and this is a little bit older picture. I have a more current one coming up at the end of the talk. But one of the reasons why I wanted to include this one is because we've had a number of students contribute to this story today that have passed through our lab that are now postdocs and doing other really great science, right? So some of this work I'm going to tell you about today is the work of Shahad Al-Qahtani, who's now a postdoctoral fellow at Howard University in Washington, D.C. And then you'll also see work by Marcel Ferreira, who's a current PhD student. There are also contributions by Dennis DiMaggio, who is a postdoc in the lab, as well as some current students. And then there's many contributions by Dr. Wansik Yao, who's a research assistant professor in my lab. The other is that we are grateful to have in our lab for a couple years at least, Gus Pendleton, who's now a microbiology PhD student at Cornell University. So all of these folks have contributed to the story today. I just want to introduce Staph aureus to you all. Perhaps you all are familiar with it. Staph aureus is responsible for 3/4 of all skin and soft tissue infections, and is responsible for 12 million hospitalizations a year. Right? And it's a big problem, not only in the US, but also globally, okay? It's not only a burden for your own health, but it's also a financial burden, right? And it's a commensal, it colonizes the back of your nose, right? So we think of it as a pathobiont. Not only does it cause single species infections, but it's also the prominent organism found in polymicrobial infections like the cystic fibrosis lung or chronic wounds that fail to close, like if you're a diabetic or if you're a diabetes patient, for example. And it's often co-isolated with other opportunistic human pathogens like Enterococcus faecalis and Pseudomonas aeruginosa. The other big part of it is it can be an occupational hazard for those that are working with animals, or if you have an animal at home, a pet, for example. So it turns out that Staph aureus can transiently colonize your pet, and so it can serve as a reservoir. You can also get zoonotic infections with MRSA. And the other big thing is it is very capable of acquiring antibiotic resistance. And so we have to think about that, right? And there was a recent projection that by 2050, it's going to lead to a really incredible number of deaths worldwide if we don't do something about these organisms. So I like to talk, so I have to warn you all now that I anthropomorphize microbes, and I blame that on my upbringing as a scientist. My PhD advisor would do the same thing, and so you are a product of your upbringing. So, but the reason why I say that it's a clever pathogen is that it is capable of colonizing essentially every surface of your body inside and out. Okay, and on the left-hand side here is just it's a figure from a review that shows you all the different types of infections where Staph aureus can colonize and set up shop and essentially make a living in and on your tissue. And the reason why Staph is so clever and can do all of this is because it has a variety of different surface-associated and secreted factors that allow it to adapt to these different environments. And if you think about it, these different locations are very different with respect to pH, nutrient availability, osmolarity, and all these other environmental factors, right? And so it makes sense that Staph aureus is going to want to adapt to these different places, right? So these are some of the secreted factors. Not only are so there's toxins, and there's also proteases that destroy. So one, they destroy complement. They also help slice and dice your tissue. And the nutrients, the amino acids that are liberated from the action of proteases, the fatty acids that are liberated from the action of lipases that are secreted, those are all nutrients that the bacterium can consume to replenish, right, depleted stores or to satiate the organism. Right? And you can imagine then that since these are all in different locations and the environments or the weather in these different environments is very different, you can imagine that the repertoire of toxins and other factors required are going to vary in these different environments. And so there's a ton of regulation that controls the production so that only the right factors are produced in that particular environment. And that's controlled by small regulatory RNAs, transcription factors, and two-component systems. And so this is one of the reasons why we're interested in understanding how virulence factor production is controlled, right? With the idea that we can perhaps interrupt that signaling, right, and prevent the production of the factors. Okay, so the bottom line here, right, is that Staph aureus, right, because it can colonize all these different sites, it's a great model for understanding adaptation in different environments, and it's also a major health problem, right? And we need to understand how it makes a living in these different environments. And I mentioned how we can think about interrupting that signaling, and this is an approach called antivirulence. And it's gaining a lot of traction, right, over traditional antibiotics that often target essential processes. Okay, and so this is just a figure from a review in 2017 that shows that the interest and the research has been climbing over the years, right? And the reason why these antivirulence strategies are gaining traction is that there's some perception that there's less selective pressure, right? And they can be specific, right? We can think about precision medicine, targeting a specific bacterium and leaving the rest of the organisms of your microbiome intact and unscathed, right? And potentially, right, they can synergize with existing antibiotics or even resensitize the bacterium to antibiotics that are no longer effective. Okay, and so we've started looking at this as well from the work that we've been doing on the basic biology of signal transduction in Staph aureus. I won't get into the data today, but if you can take a look at the paper, and I'm happy to talk with you all after about it. But one condition that is caused by Staph aureus is menstrual toxic shock syndrome. And the reason why Staph aureus causes it, it produces this toxin, TSS-T1, and it's controlled by the regulatory system that I'm going to tell you about today. And what it turns out is that we collaborated with John McCormick's lab at Western University in Canada, and a very talented postdoc in his lab, Karine Defrey, who has her own lab now, identified an already approved, FDA-approved, over-the-counter analgesic that can block toxin production by inhibiting the SAE2 component system. And so this idea that it's already been vetted for safety and efficacy, right? And now we can repurpose it to block toxin production. So this is very exciting. And so the other bit is that this drug that we've found that acts as a competitive inhibitor of the SAE2 component system for ATP, it turns out that it doesn't affect the vaginal microbiome at all. So that's a big plus for this approach. Okay, so I want to talk about virulence signaling, and I have to introduce you to kind of the founding partner, if you will, in this study, and I have to do a little bit of time travel to take you back in time to when I was a postdoc. and some of the work that I did on a transcription factor called CodY, because this is kind of the genesis of the project that I want to tell you about today. What you need to know, not that much about it, but CodY, and this is a crystal structure representation of the protein here. It's found only in the low GC gram positives, those are firmicutes. And so some non-pathogenic and pathogenic species are shown here. It's not an exhaustive list, but all of these organisms produce CodY protein. And what it does is it controls, in various ways, either positively or negatively, both bacterial physiology in terms of regulating what we call metabolism genes. So these are genes for transporters on the membrane or for enzymes that can process alternative nutrient sources, right? So in all of these organisms that share, there's a core regulon that has these metabolism genes in it, targets of CodY. And then in the pathogens, CodY has been co-opted to also control the production of virulence factors. And the idea behind this is that so CodY can actually act as a linkage between the physiology of the organism and metabolism, and allow that to inform them of when to turn on virulence factor production. In our lab, we think of virulence as just a normal homeostasis mechanism, right? It's the way to just recover and replenish nutrients that have been depleted. And that's really all virulence is. It's really just a way of bacteria feeding itself, right, when the host restricts those nutrients. So CodY is a transcriptional regulator. We also think of it as a nutrient sensor because it binds to and therefore monitors intracellular concentrations of amino acids. So these are the three: isoleucine, leucine, and valine, or ILV. And it also monitors energy status, and so it binds to GTP as well. And so at least this is just an example for isoleucine. So when isoleucine is bound, it adopts this conformation that allows it to bind to DNA and to physically block transcription from target promoters. And when that isoleucine is limiting, right, its conformation is altered, it no longer can bind to DNA, and therefore repression of those target genes is lifted. Okay? And I get this question all the time. And so as I've learned over the years, I try to answer these questions so that other ones can come up during the Q&A session. People often ask me, well, why isoleucine, leucine, and valine? Why not any other amino acid? And so if you think about it, isoleucine, leucine, and valine lie at a crossroad in metabolism. So isoleucine, leucine, and valine are some of the most abundant amino acids found in proteins. These are hydrophobic residues that contribute to protein folding and the hydrophobic effect. The precursors that make branched chain amino acids are often used to make cofactors and coenzymes, so coenzymes like pantothenate. And relevant for my talk, they're also used to generate certain fatty acids that they use to build their cell membranes as well. Okay, so by binding into and monitoring the concentrations of ILV, the cell can use CodY to monitor various aspects of bacterial physiology simultaneously. Okay, and ILV, right, as I mentioned earlier, right, the different environments are gonna vary with respect to various nutrients. And one location, right, is, it turns out, the anterior nares, right, where Staph aureus colonizes asymptomatically, right? The levels of branched chain amino acids are relatively low, and isoleucine is actually undetectable. So that environment is potentially an area where CodY activity is going to vary, and it may also allow Staph aureus to turn on particular factors to escape from that environment. The last bit, and then I'm going to talk about the real project for today, is that Staph aureus, it turns out, is what we call a pseudo-auxotroph, in that it has all the genes to produce the branched chain amino acids. But it's not like E. coli or Bacillus subtilis, if you've worked with them before, in that with those organisms, you drop them into a minimal medium, and they will very happily turn on the genes that encode the enzymes to make the branched chain amino acids. Staph aureus is stubborn now and won't do that. Even though they have all the genes, you put them into a chemically defined medium without those amino acids, and they do nothing, right? And the question is, well, why do they do that? And so it turns out that we believe that they do this deliberately so that they can pay attention to the external environment, right, during infection, and then use external levels of isoleucine, leucine, and valine to gauge where they are, as maybe as a host cue, and then as a signal to turn on particular virulence genes. And it turns out that we're not alone in this. Right around the same time that our paper came out, a group in Israel, not Hershkovitz's lab, showed that the same thing actually occurs in Listeria monocytogenes as well. Right, so this is kind of a broad strategy for pathogens to know where they are and to signal virulence. And so I'm going to skip over some work and fast forward more to the present, and just to summarize and say that in data that I don't have time to tell you about today, what we did is we played a very interesting genetic trick, and we showed that that the virulence factor production is controlled by CodY, but it's done so in a way that is indirect, and that it requires, prior to regulating these targets of CodY, it turns on the Say2 component system. And the Say2 component system, for those that are unfamiliar with two-component systems, they're made of two components. There's a membrane, integral membrane protein that's a sensor histidine kinase. So in this case, it's Say-S. And we call it a histidine kinase because upon stimulation by a signal, and the Say2 component system responds to cationic antimicrobial peptides, human neutrophil peptide that's produced and secreted by neutrophils. Okay? These are designed to be part of your innate immune response and to poke holes in the bacterial cell membrane, killing the bacterium. Okay? And so this system is actually a defense system for Staph aureus during infection. So upon stimulation, it gets phosphorylated on a conserved histidine residue. And then Say-R is a DNA binding protein. And so Say-R and Say-S interact, and then you get autophosphorylation then of Say-R. And it's the phosphorylated form of Say-R that can bind to DNA and activate transcription of downstream targets, which are the number of different secreted and surface-associated virulence factors. This is the organization of the gene at the locus. Most of this is more for your information, just know that R and S are controlled by a fairly weak constitutive promoter that's always on. So you always get basal transcription of R and S. And then there's an inducible promoter called P1, which is a direct target of, say, R, and transcribes all 4 genes. Okay? P and Q are shown here. Q is a transmembrane protein, and P is a lipoprotein. And P, Q, and S have been shown to form a complex. And this complex is needed to stimulate the phosphatase activity of, say, S towards AR to shut the system off. So think about P and Q as negative regulators of the Say system, and say S and R as the positive regulators of this system. Okay? And so baked into this is an auto-regulatory loop, right? So that you can, when the system is activated, the system can then reinforce by turning on the positive, by upregulating the expression of the positive regulators, and then also turning on the production of P and Q that keeps the SAE system from going unlimited in terms of its activity. Okay? And so just to convince you that CodY controls virulence factor production via SAR, I'll show you just a couple of experiments that we did over the years. The first is a very simple experiment where you just grow it in laboratory, Staph aureus in laboratory culture, a wild type and a CodY mutant. You take you get rid of the cells, so all that's left is the conditioned medium or the culture filtrate. You throw those onto neutrophils, and then you ask, does that culture supernatant kill the neutrophils? And the answer to that is yes. And it does so in very potently, right? So this is just showing you a range of different concentrations. And the key point here is that, so shown here, this is the wild type in open circles. And at a very low concentration of the culture filtrate, we see a low level of killing of neutrophils, but that is enhanced quite a bit, right, when you knock out CodY. And this is to simulate extreme nutrient deprivation, right? And those virulence factors those genes are de-repressed. And if you make double mutants and look at the SAR single mutant, you can see that the effect is via the SAE two-component system. And similarly, if you go in vivo in two different, if you look in a skin and soft tissue infection model, we can see that in black, this is a wild type cells that have been challenged, or mice that have been challenged with wild type Staph aureus cells. They cause skin and soft tissue infection, dermonecrosis, and that dermonecrosis is enhanced in a CodY mutant, and this depends on SAE, and one of the targets of the SAE two-component system, which encodes alpha toxin. And we know that this is through secreted toxins in that the bacterial burden actually doesn't change between wild type and a CodY mutant, but rather it's more of the secretion. And so I wanted to just, again, summarize by saying that we did, so a lot of the work that was performed by a very talented graduate student in the lab, Kevin Malinik, who's actually a junior investigator now at Fort Detrick up in Maryland, working on Francisella tularensis. He and a research assistant in the lab, Nick Waters, did some really nice work to show that CodY directly and indirectly controls the expression of the SAE locus. Okay, and so this allows the SAE system to be regulated as a function of nutrient availability, as well as some other factors that I'm not talking about today. But if you're paying attention, you might be thinking, oh, that's funny. That looks different on my screen versus here, but it's the wonders of technology, right? But it works, right? You probably are scratching your head here, right? Because I just said before that CodY represses the expression of the SAE system. It turns out it's at this P1 promoter, right? And so what this is doing is in response to nutrient deprivation, CodY is actually de-repressing this locus, which actually encodes these inhibitor proteins. So what the heck is up with that, right? Why would you, it's kind of a paradox or a conundrum. CodY is actually turning on virulence factor production by turning on the negative regulators of the SAE system. That doesn't make any sense to you. So we wondered whether or not that perhaps CodY's control of the SAE locus is twofold. Maybe there's a transcriptional effect, but then maybe there's some other effect. And I'll get to the other effect in a moment. But perhaps the effect on the P1 promoter is to make sure that SAE activity doesn't go unlimited. And so this was, so to test this, Shahad in the lab at the time, she did a very simple experiment. What she did was, first she compared wild type to a CodY mutant. And so she used a direct target of the SAE system that's also controlled by CodY. It's one of the virulence factors of Staph aureus thermonuclease. And it's what she did is she hooked up the promoter to nuke to GFP so she could follow the activation of the nuke promoter. And she looked and she noted that in wild type cells, expression is relatively low. Again, this is during laboratory growth. However, when she knocked out CodY, we see the hallmark of a CodY negatively regulated gene, and that it is de-repressed, right, under the same conditions. But then what she did was that she deleted the say locus, and then she just expressed ectopically R and S using that constitutive P1 promoter. So got rid of P and Q, got rid of P1, right? And what does she see? She sees that you get the same effect, right? So P1 has nothing to do with activation of the say system by CodY. So what she's done is she's shown that transcriptional regulation and activation of the say system, both by CodY, can be separated and uncoupled, okay? Which means that there's something more and more interesting going on, right? And this is just to show that we can look at promoter activity for nuke, but we can also look at secreted nuke activity, right, in culture supernatants. And we can see, again, the data mirror the transcript, the promoter data, okay? And so we probed a little bit more. And what we found was that in CodY deficient cells, Wansik Yao in the lab showed using an approach called phosphag electrophoresis. So this is a chemical additive that you can add to your protein gels. And what it does is it slows the migration of phosphorylated proteins. And so this is just using recombinant protein. And you can see that, so we're using say S with a tag on it, right? And then we, remember that in the presence of ATP, it can get phosphorylated. And then in the presence of say R, you have phosphoryl transfer to say R to give say R phosphate. And you can see that we can separate say R phosphate from say R, at least using recombinant proteins. And it turns out it works using cell lysates and antibodies as well. So we use two different strains of Staph aureus or two clinical isolates. One is the dominant clone in North America, USA 300, and then a bone infection isolate as well. And in both cases, we can see this is the phosphag and Western blotting that we see, and then this is the quantification of the data. And in both cases, we can see that say our phosphate levels are increased in a CodY mutant. And it turns out that two-component system kinases and response regulators are often very specific, and therefore you don't get cross-talk often between these different two-component systems, which means then that say S activity is the sole determinant for say R phosphorylation, right? And so what this means then is that in the CodY mutant, say S kinase activity is heightened or increased relative to wild type. And we can use more traditional radioactivity assays to follow this over time as well. Okay, so just to recap so far, right? So what I've told you is that CodY and the branched chain amino acids are important for controlling bacterial physiology as well as virulence factor production in gram-positive bacterial species. In Staph aureus, what I've shown is that CodY upregulates pathogenic potential via the say system. And hopefully I've convinced you that CodY's role in controlling virulence via the say system is actually much more interesting than transcriptional control, right? So the question then is how then is this activation or control of activity occurring? And so we went back, Shahad and Gus and I were talking. And we noted something very peculiar about this say S kinase. So typical kinases for two-component systems have an extracellular domain that binds to a ligand or your signal, okay? And then that binding event causes a conformational change which transduces that signal intracellularly, and that elicits a cellular response. But it turns out that say is part of a growing number of kinases that we acknowledge actually lack that large extracellular domain. Rather, it actually has a tiny little 9 amino acid peptide linker. And this is just for the monomer. It turns out CS is a dimer for its function, right? But here, you can see that if the true signal is a human neutrophil peptide, there's very little information provided by a 9 amino acid linker that's going to confer binding or specificity, right? So how that all works, we're still trying to figure out now. But what this means then is that perhaps that any perturbation in the bilayer, the plasma membrane, may trigger, say, activity. Okay? And so that makes sense, actually, or is still aligned and consistent with the known role or cue for, say, and that is human neutrophil peptide. Remember that I said that these are cationic antimicrobial peptides that perforate the cell membrane. Right? And so the cell is perhaps sensing this disruption in some way. So could it be then that lipids, right, within the bilayer can act as a second messenger? Right? So the first signal, right, may be, human neutrophil peptide, right? And is that now converted to some other message, right, a second messenger that can perhaps bind to, say, S and affect its activity, okay? And so this is just a model that they came up with, right, whereas perhaps in the presence of some lipid component, say S is on, and then in the absence of that lipid component, say S is off, okay? And so this is where teaching and research come together, right? They actually are not in conflict with one another. They both inform one another. And I teach this, and this is actually a slide that I tweaked a little bit from my lecture on signal transduction in biochemistry for undergraduates, right out of Leninger biochemistry. And we talk about G protein coupled receptors, right, that are activated, and in response, to their ligand binding, right, they go on and activate an enzyme that generates a second message, right? And in this case, the one that I'm trying to draw parallels to is certain GPCRs actually activate phospholipase C, which cleave, right, a lipid to diacylglycerol and inositol triphosphate. Okay, and the whole point of this is amplification of a low-level signal, right, to elicit a response, right? So again, I remind you then again that perhaps maybe depending on CodY status, right, perhaps, right, maybe, I alluded to this before, perhaps fatty acids that are derived from these amino acids might be part of the answer and part of this second message. So I have to tell you a little bit about branched chain fatty acids, which are, it turns out these are the predominant fatty acids that are components of the glycerol phospholipid, right, in staphylococcal membranes. So if you go back to biochemistry, in undergraduate days, you learn that a glycerol phospholipid has a glycerol core, 2 acyl groups, and a head group, okay? And those acyl tails can be a variety of things. They could be saturated fatty acids. They can be, and it turns out they can also be branched chain fatty acids, right? It just depends on what is used to build the fatty acid initially, okay? Some organisms also have unsaturated fatty acids, kind of like you and me. You and I have unsaturated and saturated fatty acids as part of our phospholipid bilayer, and that helps maintain fluidity. So it turns out Staph aureus cannot make unsaturated fatty acids, nor can they desaturate saturated fatty acids. So it turns out that they rely on these branched chain fatty acids, right? And they can be named in different ways depending on where the methyl group is, right? And they rely on those in the same way that we rely on unsaturated fatty acids to fluidize the bilayer, right? We learned that unsaturated fatty acids have a kink, and that prevents packing. Well, the methyl group, that bulky group, also helps prevent packing and help fluidize the membrane. Okay, so it turns out that they rely on those. I will say that Staph aureus can utilize unsaturated fatty acids and incorporate them from the host during infection, but it turns out that that's insufficient. Because if you take, so in green here, we've challenged mice, a skin and soft tissue infection model with wild-type Staph aureus, and you can see that dermonecrosis that we saw earlier. And so we quantify the number of bacteria in those lesions, right? But again, if you take say mutants, you can see that they don't form these dermonecrotic lesions. But if you also take a strain that cannot make the branched-chain fatty acids, if they're a branched-chain fatty acid oxytroph in the host, they're dead in the water, right? So that means that even though they can acquire host fatty acids, they can't play the same role, right? So there's a particular dependency on branched-chain fatty acids. So what do we know so far, right? We know that CodY influences, say, activity, and we know that CodY controls virulence, various targets. One of the things I slipped by is that it also controls the genes for branched-chain amino acid synthesis. But what we don't know is, does CodY influence branched-chain fatty acid synthesis? And is it that branched-chain fatty acids influence, say, activity? So to begin to answer that question, Gus and Shahad and then Dennis also teamed up together, and they started asking a series of questions. And they looked, and so what we did is we first isolated membranes from various strains, and then performed gas chromatography fatty acid methyl ester analysis, or GC-FAME, right? This just quantifies relative levels of fatty acids in the membrane. And what we can see is that if we look at the branched chain fatty acids, particularly the one derived from isoleucine, we can see that the abundance in the membranes goes way up in the CodY mutant. That branched chain fatty acid oxytroph, right? It's an oxytroph. We've played a couple genetic tricks that I'll get back to you in a moment on, but if so that we can grow them in the lab. But it turns out if you deplete cell membranes of these branched chain fatty acids, right? They have, you can see that by GC-FAME, you see that they're quantitatively reduced. And also we see that the membrane fluidity decreases, right? So membrane rigidity goes up. This is fluorescence polarization that was performed by Vineet Singh for us as part of that story, okay? So far, there's a correlation between branched-chain fatty acid levels and CodY and, say, activity. Oops, and that didn't turn out well. But hopefully you can appreciate this is, say, our phosphate levels. So say CodY, we saw this earlier, higher, say, our phosphate levels, implying, say, as kinase activity is increased. And then in the branched-chain fatty acid oxytroph, we see low levels. So again, correlation between high activity of, say, and high branched-chain fatty acids, low activity of, say, low branched-chain fatty acids, okay? So then this is where we teamed up with Marcel Ferreira in the lab. So simultaneously, and this is a really cool story, I don't think we'll have time to talk about it today, but I'm happy to share it with you later. Marcel has been looking at, what we've done is we've discovered a second pathway to make these branched-chain fatty acids. So while she was working on this, she had discovered a mutation that I actually showed you over here. So this is just the designating that this is upregulating the second pathway. And in this strain, right, the important bit is that we can grow the cells, but they have very low, say, activity. Okay, so it's a tool that we can use, and I'll leave it at that for now. So Shahad decided to make use of that tool and ask, what happens in that strain when you knock out CodY? So again, the cells have very low branched-chain fatty acid levels, right? And so CodY mutants in that background do not have say, activity. And this is in a rich medium where the levels of exogenous fatty acids are very low. And it turns out that this enzyme MBCS that Marcel discovered is important for converting these carboxylic acids, right, to the acyl-CoA, which are used to make the branched-chain fatty acids. So Shahad made use of this strain, and what she saw is that there was a strict requirement for CodY to do its job and to activate the SAY system, or to control the SAY system. It had to have fatty acids at its disposal, right? The branched-chain fatty acids, and that was specific in that really, and you can, so the important bit to compare here is this bar here, right? So in a CodY mutant, in that strain background where branched-chain fatty acid levels are very low, right? You have very low SAY activity, but then when you provide the precursors for the fatty acids, right, for the ones derived from isoleucine and the ones from valine, right, in the checkered boxes and the gray boxes, those now restored SAY activity. Okay. Now it seems it's specific in that the leucine derivative doesn't do it. Okay. So at the end of the day, what we see is CodY requires branched-chain fatty acids to upregulate kinase activity. And so Shahad then wanted to explore this a little bit more mechanistically. Right? How does that occur? So then we went back to the data that Nick Waters, when he was a research assistant in the lab, collected using RNA-seq. And what he saw is when we looked at targets of CodY, and what we saw is that CodY controls, again, branched-chain amino acid synthesis, but then also the genes that encode the permeases for the uptake of branched-chain fatty acids, branched-chain amino acids from the environment. Remember that Staph aureus is a pseudo-auxotroph in that it depends on external ILV, right, often to satiate its needs for branched-chain amino acids. And so they're highly upregulated in a CodY mutant relative to the wild type. So then Shahad then asked the question whether or not CodY actually is controlling SAY activity by controlling the synthesis and the import of branched-chain fatty acids. And the reason for that, again, is the shared precursor, the alpha-keto acid. So this was the model that Shahad had. And so what she did was then, again, looked at promoter activity of a SAY-controlled target. Right? It's also controlled by CodY. Again, here's that nuke promoter. And again, CodY mutants show heightened SAY activity. If she made individual double mutants where she knocked out the genes that encode for these permeases, BR and Q, 1 and 2, or she knocked out CodY and also the ability of Staph aureus to make branched-chain fatty acid precursors, she found that there was no effect. But it turns out that if she blocked de novo synthesis of the precursor and then also blocked isoleucine transport specifically, that's through BR and Q2, she saw, although it looks mild here, it is a significant difference in SAY signaling. Okay? So it does appear then that CodY seems to upregulate SAY activity by controlling the generation of the abundance of these precursors in the membrane. She went on to show then that if you look at the branched-chain amino acids, and these are two different forms of it, in that same mutant, right, that we just saw, that the reduction in SAY activity, in red bars, you can see that the fatty acid abundance using GC-FAME, again, is low. And this is complementable by bringing back that transporter on a plasmid. Okay? So then she decided to go one step further. She decided, well, if this is how CodY does it, then we should be able to uncouple this from CodY altogether and just take wild-type cells, overexpress BR and Q2, so therefore increase perhaps the influx of branched-chain amino acids into the cell. And that, if we're right, this should increase SAY activity. So again, she used promoter fusion. This is a slightly different one, but it does the same thing, and it monitors SAY activity. And again, these are our controls, right? So CodY mutant, you see upregulation of SAY activity read out by this promoter fusion. So this is largely say-specific, so you see that the activity is low in that background. And then in wild-type cells, if you express BRNQ2, right, you can see in a dose-dependent manner, when you increase expression of BRNQ2, you get an increase in, say, promoter activity, okay? And then this is, and I'm not showing it here, but this doesn't happen in vector-only control. And again, you can see there are some mild differences, increases in branched-chain fatty acid levels as well in the membrane, okay? And again, for some reason, these particular graphs don't show up well, but I hope I can convince you that if you look specifically at kinase activity, or using that FOSTAG trick that we did before, I hope you can see that in wild-type cells, when you express BRNQ2 in these checkered blue bars here, you can see that kinase activity goes up, and you see it's very similar to that in the CodY mutant. Okay, so the model appears to be correct then, that we upregulate, say, activity by increasing the abundance of a particular lipid in the membrane. Okay, so just to recap, right? So CodY deficient cells have an increase in branched-chain fatty acids that are important parts of the lipid bilayer. And she showed through genetic analysis that synthesis of isoleucine specifically is important to lead to that particular branched-chain fatty acid. I slipped it past you, but that branched-chain fatty acid auxotroph is part of a multi-enzyme complex that synthesizes these precursors for fatty acid synthesis. And so by blocking that activity, right, cripples the activity, and so this is actually really interesting, and perhaps we can use this, maybe there's some therapeutic value here. Again, perhaps we can exploit this. So just as a final summary, I hope that today, right, you're convinced that branched-chain amino acids are important signals for bacterial physiology and virulence. CodY controls metabolism and virulence gene expression, and CodY passes that nutrient depletion signal to, say, through a second messenger, right? Or at least we like to think of it that way. Branched-chain fatty acids modulate, say, activity. And this story really shows, this is the first time that branched-chain fatty acids have been shown to control virulence in a post-transcriptional mechanism. Okay, so again, why would these lipids be good second messengers? Again, synthesis is upregulated as a result of a primary signal, in this case, nutrient limitation. Right? Again, we talked about this idea that say is monitoring membrane status. Right? So it's well positioned to think about it, to look for damage, right, or perturbations. So this would require remodeling. Right, it turns out that CodY also controls the genes for beta oxidation, so allowing Staph aureus to degrade the fatty acids and therefore resynthesize them. Right, so there's additional support for this. And again, perhaps, right, if we were to target, say, as an antivirulence strategy, perhaps this might resensitize or sensitize the bacteria to membrane-targeting antimicrobials, right? Okay, so very quickly, because I'm out of time, I just wanted to mention to you that we're pursuing this actively. We're grateful for funding from the NIH, specifically right around this time, right, to pursue our questions. And what we're trying to figure out is how, on the molecular level, this lipid is working, right, as a second message. And so we're testing between direct and indirect models. So the first one is whether or not the branched-chain fatty acid, as part of the glycerol phospholipid, actually buddies up against and makes contact with, say, S. And so does that somehow trigger, say, S activity? And to do that, we're actually poised to do that, making use of some tools. Before, I mentioned how Marcel had discovered this new enzyme that allows the salvaging of the carboxylic acid precursors that allows us to bypass the de novo pathway. And so we've teamed up actually with Kalal Gupta at Yale University, who's actually a eukaryotic membrane biologist that studies signaling complexes as part of the synapse in neuronal cells. And he has a method for actually using chemical polymers to stabilize and purify signaling complexes right out of the membrane, intact. And what he can do is do native mass spectrometry and lipidomics to look at the lipid content directly surrounding the kinase in the membrane, and can also look at oligomeric state. And so these are just data to show that we can take, say, S and we can tag it, and it complements. And so we now have a way of pulling out the signaling complex right out of the membranes using an epitope tag. And so this is some of our preliminary data using some of these chemical polymers to stabilize. And what it looks like is that say S looks like a monomer under conditions where activity is relatively low. Okay? And so this is Kalal, and he's actually done this before with a bacterial transporter, and he showed previously that cardiolipin, which is another double glycerol lipid in the membranes, it was important for oligomerizing that transporter. And so his idea is that perhaps some of these signaling complexes dimerize very weakly in the absence of the particular lipid that they need. So we're pursuing that. The other last bit that I'll tell you is that it's possible that it's indirect in that it's not direct lipid binding, but perhaps, as I said before, these lipids are important for optimizing fluidity in staphylococcal membranes. So is it possible that these lipids are important for allowing a negative regulator or a positive regulator to diffuse and make contact with Staph aureus, say S, and therefore activate or rather just broadly control its activity? And so we've found that, again, say S and say Q form a complex, and so we're going to look to see using FRET whether or not that this interaction is branched-chain fatty acid dependent. And so so this is just an example of this where we can use FRET. And so say S is labeled with GFP, say Q is labeled with M-cherry fluorescent tag, and that only when the two are together do we see energy transfer and FRET. The other is that we see a protein called FLO-A, which is conserved in all domains of life. And this is a scaffolding protein that organizing signal complexes in eukaryotes all the way down to bacteria. And it turns out that say S does have, in our preliminary bacterial 2 hybrid assays, seems to interact with FLO-A. So TBD on that. So I'm out of time. I just wanted to wrap up and, again, mention those that have done the work in the lab. This is a more current lab photo. We went bowling recently. So I've just highlighted previous members and current members that have contributed to this work in bold. Our collaborators are funding. And with that, I'll just mention, because I'm co-director for graduate studies, I wanted to plug our graduate program. If you're interested in earning a PhD, if you haven't already done so in Washington, D.C., check us out, right? Applications are due on December 1st. So thank you very much again for the opportunity to talk.
Hey, thank you. Very, very interesting talk about here in the corner. Yep.
Oh, there you are. Yeah. Sorry. Yes.
So I'm curious in terms of these models of the direct versus indirect, right? So I have no idea over what range staph will grow in terms of temperature, but if you were to drop temperature and presumably increase branched chain fatty acids, would you then see a subsequent activation of this pathway?
Yeah, that's a very good question. We've thought about this. So a couple of thoughts on that. So in other organisms, right, there's definitely been a lot of work done to show that as you drop the temperature, branched-chain fatty acid levels increase, right? That's homeoviscous adaptation. The challenge with Staph aureus is we could go lower, but we're trying to stay physiological. And the temperature range, really where Staph aureus is relevant, is core temperature 37 degrees Celsius, when you have a fever, which is around 40 or so Celsius, and then on your extremities, which is around 32. And that delta has been, that temperature range has been explored by a colleague, Ronan Carroll's lab at Ohio University. And they didn't see any change in a lot of the genes that we're paying attention to here. So we don't think, at least in that particular direction, is going to be a little challenging for us to probe.
If you could go lower, even though it's not physiologically relevant, it might separate the two, right? So because you would keep the viscosity of the elasticity of the membrane, right, as you drop, because you're going to try to compensate by increasing the branch chain. And so if it doesn't activate, that would potentially tell you that it's not the physical properties of the membrane, but it's more a direct interaction.
Yeah, absolutely. You're right. Thank you. Yes. Hi.
Can you remind me of why you think the regulation through the membrane of SAES is a regulation of kinase autophosphorylation rather than kinase phosphatase activity?
Yeah, that's a good question. So all I can say is that in the data we've seen increases in kinase activity. Now, whether that is because you are suppressing phosphatase activity and making it more kinase dominant, so those details really haven't been worked out yet. We're not clear. It's possible that the way this works is that we're somehow inhibiting phosphatase activity, and therefore kinase activity goes up indirect, I guess, as a result of that. Is that sort of?
Well, I guess I'm wondering whether it could be just the basal level of phosphorylation of SAER that increases because of an inhibition of SAE.
I see.
Phosphatase activity. The non-specific phosphorylation.
Yeah, I have to think about it some more. I'd be happy to talk more with you afterward, maybe. Yeah, there's a lot to think about.
Hi, Shaun, here in the middle.
Hey.
It was really fun. Thanks for sharing on this. So I'm going to ask you a speculative question.
Okay, I love speculation.
So I guess these are all kind of like the same thing, right? So I'm wondering if you could elaborate a little bit about, say, aureus is regulon, and whether this two-component system exists in non-pathogenic Staphylococcus. The reason I'm asking that is because Staphylococcus can, exist in environmental, environments which don't have, branched chain amino, branched chain fatty acids, right? And so wondering like why, if this is kind of common across different Staphylococcus, what might be the selective pressures that keep the system, right? Or not, or maybe, basically, does this exist in non-pathogenic Staphylococcus?
Yeah,
that's really what I'm asking. If it does, why does it exist?
Yeah, no, that's actually, I have to admit, I don't know, right? So there are, for example, Staphylococcus xylosis or carnosus. Some of these Staphylococci are used in food microbiology. And so now you've got my curiosity, because I don't know whether or not they actually have a say system. I'd have to go back and look and see. So I'm gonna hold off until I look, and then I'll reformulate my speculation. We can talk more later.
Thank you.
Thank you so much.
Thank